We have learned that the cell is not just a bag of flimsy protoplasm. Instead, it is an extremely complex structure functioning very much like a factory, making vital components such as proteins, carbohydrates, lipids, and nucleic acids are all important in carrying out the functions of life. Now, today we will discuss the cell membrane. The cell membrane is a vital component of the cell. The cell membrane, often referred to as a plasma membrane, is a selective membrane in that it allows certain material to go through and certain material not to go through. It's a complex dynamic structure with a purpose to control movement of substances into and out of the cell. It is important to note that all cells have this structure, be they animal cells, plant cells, bacteria, and so on. Now let's take a close look at the structure of the cell membrane. If we look at an overview, as you can see in the slide, the cell membrane is made up of the basic component, which is a phospholipid bilayer. Embedded within the phospholipid bilayer are molecules of cholesterol. And further and most important, embedded within these structures are protein molecules, and there are a number of protein molecules that we will discuss in a moment. Superimposed on the protein molecules are sugar molecules, referred to as the glycocalyx. Now let's look at the basic component of the cell membrane, the phospholipid layer. If you remember, we discussed the phospholipid molecule having a polar and a nonpolar structure. The polar head is hydrophilic, and the nonpolar head is hydrophobic. Now, as you can see, the lipid bilayer meaning two layers of lipids, are organized in a fashion so that only the hydrophilic side turns towards the inside of the cell or towards the outside of the cell. Now look at this very carefully and understand how this works. Now we get to the important protein molecules that are superimposed or embedded within the cell membrane. We have several. We have structural proteins, we have recognition proteins, we have communication proteins, and we have transport proteins. Now let's start with the structural proteins. This is pretty obvious in that the structural proteins are designed to give the cell shape. The structural proteins are connected to the cytoskeleton within the cell itself. Now, as we move to the right, you see the recognition proteins. Now, this is an extremely important protein. The recognition protein serves as a signal to the immune system. And basically, it says that I am friendly, please do not eat me. You have to understand that the immune system is designed to go around and eat anything that looks foreign to it. The recognition protein is designed to tell the immune system that I am part of this system, I am not to be harmed. Now we'll look at the next protein, which is involved in communication. Now cells talk to each other. They talk to each other for the purpose of communicating and determining what function a given cell should carry out at a given time. Now, all of this activity is carried out through signals that are sent to the communication proteins. And finally, to your right, we have transport proteins. We will talk about transport proteins in a little more detail in a moment, but the transport proteins, as you might expect, are designed to facilitate the movement of molecules into and outside of and out of a cell. Before we can discuss the movement of material into and out of a cell, we have to understand the concept of diffusion. Now, we have simple diffusion, which is the movement of molecules from an area of great concentration of molecules to an area where the molecules are less concentrated. When you put a drop of ink into water, we witness simple diffusion. Now, another type of simple diffusion is osmosis. Osmosis is a form of diffusion where we look at the movement of water molecules. The water molecules will always move from an area of high concentration of water to an area where the water is less concentrated. For example, when water moves from an area where it is very pure to an area where it is very salty, the salted area has a very low concentration of water. So water will always move from an area of high concentration of water to an area where the water concentration is very, very low. Now, let's look again at simple diffusion. As you can see in the example I mentioned before, when we drop a concentrated amount of ink into a solvent like water, we see the molecules disperse. They're actually moving from an area where the molecules are highly concentrated to areas where the molecules are not there or not concentrated at all. Eventually, you're going to get an area of equilibrium. So this is basically simple diffusion, the movement of molecules from an area of high concentration to an area of low concentration. Now, when it comes to osmosis, it's a little bit more complex process. Osmosis is responsible, for example, for the movement of water from the roots all the way through the top of a tree. Okay, the illustration that you see shows two vessels separated by a semi-permeable membrane. You might look at this as two cells that are next to each other with one membrane between the two. If you have water on the left side, the water is very, very concentrated with the water molecules. On the right side, we're going to add a solute to it. Therefore, the water on the right side suddenly becomes less concentrated. Yes, the salt is higher concentration, but the water is actually less concentrated. So, as we go to the B part, you see that the water is going to move from left to right. Keep in mind, the solute or the salt cannot pass through that semi-permeable membrane. So the water will move from left to right. As you can see, the water will now rise in that vessel to the right. So, in osmosis, water is moving from an area of high concentration of water to an area of low concentration of water. Now, why does this happen? Well, if you look carefully, what really is happening is the water molecules are surrounding the sodium chloride, which is a solute, surrounding the sodium chloride and basically tying up the water molecule itself. Therefore, it stays to the right, and eventually all of the water molecules pretty much will go from left to right. So when we look at osmosis, we have to understand how osmosis functions in the real world with respect to a cell. Remember, a cell has to maintain homeostasis, maintain its balance internally and externally. And one of the balances it has to maintain is maintain the balance of salt concentration and maintain the balance of water concentration. Now, we often describe cells in these conditions in the following fashion. We refer to them as isotonic if the internal and external cell has the identical concentration of solute. That's an isotonic condition. A hypertonic condition is when the external cell has a higher concentration of solute than the internal cell. And in the hypotonic condition, the external cell has a lower concentration of solute than the internal cell. We have all witnessed this reaction when we take some lettuce and we try to get it crispy. What do? We put it into some clear water. By putting it in clear water, which is a hypotonic solution, we're actually allowing the water from the outside to move into the cell and get the salad crispy. If we then add salt and pepper and everything else to it and come back the next day, what do we see? We see everything is wilted because we have changed the conditions. We have turned the external cell environment into a high salt concentration, forcing the water from the cell out and collapsing the cell. Let's look at this example in more detail on the next slide. Here we are looking at an animal cell, a plant cell, and we are looking at these two cells under isotonic condition, hypertonic condition, and hypotonic condition. Notice what's happening to the cell under these conditions. Under the hypertonic condition, the cell will shrink up. Under hypotonic condition, the cell will expand. And under isotonic condition, everything stays cool. In the real world, in salt water, in fresh water, a cell has to adjust itself to the environment and maintain its structural integrity. So now let's get back to the main point of this lecture: movement of molecules across the cell membrane. We've discussed the concept of diffusion, we've discussed the concept of osmosis, but now let's look at movement of small molecules across the cell membrane, and we can break it down into two categories. One is passive transport, and the other is active transport. In passive transport, we are basically looking at simple diffusion and facilitated diffusion. There's no energy required in movement of molecules across a cell membrane in passive transport. In active transport, energy is required. Nothing will move through a cell without the utilization of energy. So now let's look at passive transport and active transport on the next slide. To your left, in simple diffusion, you can see that the molecules in blue on top are highly concentrated. Therefore, they will tend to move towards the opposite side of the membrane by way of simple diffusion. Now, these molecules will move from an area of high concentration to an area of low concentration. This happens all the time, and this is the first example of passive transport. In another example to the right, you see facilitated diffusion, which is also passive transport. But in this case, the little red molecules cannot get through the membrane on their own. Yes, they're highly concentrated on one side and the low concentration on the other side. The only way they can get through is through a protein that facilitates the transfer of those molecules. But this does not require energy. The protein simply has a channel for those red molecules to move through, and eventually the molecules will move through the process of diffusion into the cell. Now finally to the right, we'll look at an example of active transport. In this case, something else is going on. We have a protein that's embedded within the lipid layer, But this protein, yes, it also facilitates the diffusion of the blue molecules. But the only way that happens is if energy provided by the molecule ATP, that you will learn a lot more about later on when we deal with a chapter on energy, adenosine triphosphate. This molecule provides the energy for the protein to basically pull those blue molecules through and deposit them within the cell. You notice that the molecules on the outside of the cell have a lower concentration than the molecules on the inside of the cell. It doesn't make any difference what this concentration gradient is. The molecules will be pulled in because the energy from ATP causes the protein to pull those molecules through. Now let's look at a more complex example of active transport. And this is the example we will use is the sodium-potassium pump. Sodium and potassium are important ions that have to be balanced in a fashion that it is most beneficial to the metabolism of a cell. Now, as we look at the membrane of a cell, looking the top being the outside of a cell, the bottom being the inside of the cell. Here we're looking, of course, at a cell membrane with transport proteins present. If you start from the left, you see that the protein is open. And these little proteins act like tiny little pumps. On the left, you see that two sodiums are bound to the protein. Another sodium is getting ready to hook itself to a binding site. And when this happens, energy from ATP comes in and pinches that protein. And when it pinches that protein, the three molecules of sodium are shoved outside of the cell. When this happens, potassium, which is on the outside of the cell, finds binding sites on the protein to attach itself. And when it does so, the configuration of the protein molecule changes and it snaps shut on the top, releasing the potassium. So what is happening here is that for every 3 sodium that are pumped out, 2 potassiums are pumped in. The reality is that membranes have tiny little pumps, and these pumps are little protein molecules that open and shut, and this opening and shutting is carried out by way of ATP providing energy. Now, sodium-potassium pumps are important in a number of physiological activities, and it is important that you understand how this works. Now, so far, we have looked at the movement of small molecules, maybe even some macromolecules across the cell membrane. However, in the movement of large material into a cell, we have a number of other processes that play a role. A cell will carry out what is referred to as pinocytosis. It will carry out endocytosis and exocytosis. Basically, it's the movement of big chunks of material into a cell and out of a cell. A cell sometimes also will work through a receptor-mediated endocytosis, and sometimes a cell will just go and engulf a complete bacteria all at once, and we refer to this as phagocytosis. Now, let's discuss each of these for a moment. Now, we're looking at a cell, and in this case, the cell is going to carry out the process of pinocytosis. As you can see, the green molecules are on the outside of a cell. They come in contact with the membrane. A vesicle is formed. bringing in the material to be utilized by the cell. The formation of vesicles is a common process that cells use to basically engulf chunks of liquid material. Now, this can also be reversed. A vesicle can form, for example, a vesicle can form with the production of protein, and this process gets reversed and material is released out into the extracellular environment. Now, in exocytosis, what is happening, a cell is producing large amount of macromolecules, perhaps in the Golgi apparatus. These macromolecules are encased in a vesicle, and this vesicle will fuse with the membrane of the cell and release these macromolecules, in this case proteins, out into the extracellular environment. This is referred to as exocytosis. And of course, the reverse of this would be endocytosis, where material from the outside is pulled, a large material from the outside is pulled in, forming a vesicle and bringing it into the cell for use. Another process where a cell will specifically bring in material into its structure is referred to as receptor-mediated endocytosis. Now, an example of this might be the relationship between an immune cell and a virus. The immune cell will have special receptors present that will recognize that virus. When the immune cell comes in contact with a virus, these special receptors will bind to the virus, engulf it, bring it in, and neutralize it. Now, there are many examples of receptor-mediated endocytosis that we cannot go into, but this is a very important process by which cells bring in material into its internal structure. Now, finally, we get to a process called phagocytosis. Now, in phagocytosis, we are looking at a process by which an amoeba would engulf a complete organism and eventually digest it and utilize its nutrients. Or you might look at a white blood cell coming in contact with a bacterium, and the bacterium being engulfed and encased in a vesicle and eventually digested and destroyed, and so on. So all of these processes that we discussed are important in the relationship, in the way cells go about bringing material in and, of course, getting material out. Okay, so what have we learned? We have learned that the cell membrane is a vital structure on a cell. It controls goods coming in, and it also controls goods going out. As the cell membrane also is important in signal transmission. And the cell membrane gives the cell shape and form. So with all of this, we have a better appreciation for the overall structure of a cell and how it functions to maintain the processes of life.